Hi, my name is Beau Alward. I'm a 2021 Beckman Young investigator. I run a lab at UCLA in the Integrated Biology and Physiology department. And I'm really excited today to do this video as a part of being a Beckman Young investigator to do a walkthrough of what I've accomplished due to the support of the Arnold and Mabel Beckman Foundation, and such an inspiring group of people from history as a part of the Arnold and Mabel Beckman Foundation, and the support I received while there. So I'll get started. I'm going to share slides to discuss really what the foundation has meant to me as a scientist, as an individual, being able to grow. And it's going to be focused on my goal as an investigator, a Beckman Young investigator, a BYI, to develop a model of social behavior. It all started for me really before I became affiliated with this foundation. Where I wanted to pursue the unknown. This is where it's always been for me, and really starting out on a path that nobody has pursued. That's what has excited me most. I'm a neuroscientist, and what that typically means if someone wants to study neuroscience is starting out research in mice. That seems to be a very popular approach, or in rats. That has never really caught my eye. And this led to sort of ahead when I wanted to understand the mechanisms of autism. This was something that was near and dear to me. Before I started graduate school, I actually worked with children who had autism, trying to enhance good behaviors in them, to help them, thrive in the world. And I just kept wondering what was going on that led to this. And this um, led me to want to pursue questions related to social behavior because that is one key thing that could be lacking or perturbed in those with autism. And really, the discovery when looking into it and hearing from the field is that autism is very complicated. And one way to gain insights into this is to study mechanisms in diverse ways, in diverse species, gathering information from species that are different that may actually have a convergence of mechanisms. So before I even started a postdoc, I actually proposed to the NIH to study a highly social cichlid fish to understand autism. And I remember speaking on the phone to a program officer at the NIH, and they basically laughed at me. And I will never forget this moment because it led me to the Arnold and Mabel Beckman Foundation when I was a postdoctoral fellow at Stanford University. So I have been a part of this foundation since 2017. This is where I found my home for pursuing my research questions. And this is part of the goal, which is to fund new ideas that aren't developed enough to be federally funded. And I received this postdoctoral fellowship in 2017 to study cichlid social behavior. And it was when I became a part of this foundation that I started to understand the history of Arnold Beckman himself, and how it's so inspiring, and how it started to um, give me an example of something I wanted to pursue myself, but I really not knowing what was out there about who have done this before. Arnold Beckman really laid out a path for me and those who are part of the foundation as well, where he was trying new things for the good of humanity in the terms of developing systems to understand chemicals that could lead to improvements in health and developing medicines and knowing more about the nature of biology. He demonstrated the importance of working in a team. That is something I've kept with my lab ever since I started it. And something that is very important to me is allowing great people to come into my lab and reach their potential. And Dr. Beckman really laid this out for me, and that is what led me to want to develop to the cichlid fish as a model to understand the molecular and neural mechanisms of social behavior. And here is this model. You can look and see that this fish is brightly colored. This is a dominant fish of the species I study. And this is a subordinate or non-dominant fish. And these fish that are dominant, they perform territorial behaviors, they're reproductively active, and they're pursuing their females, okay? They're doing this with these behaviors, but the key factor of this is understanding these mechanisms, right? Is the dom males have high androgens, such as testosterone and estrogens, and these have been hypothesized for so long to regulate behavior in these species. And the non-dominant or subordinate males are really the opposite of the dominant individuals. You could look at their physiology and find that dominant individuals, they're reproductively active, and you can see that in the size of their gonads, and then subordinate ones have smaller testes. And here's what's key about this for a neuroscientist. They have an altered brain depending on social status. It's in these gonadotropin releasing hormone neurons that are larger in the brains of dominant individuals and smaller in subordinate ones. And so for decades, this information was known. It was laid out by researchers ever since the early 80s. And when I started on this work, I wondered if there's social control of this, if they're dominant or subordinate, based on what's around the environment of these individuals, I wanted to dissect how that is occurring. In other words, what underlies their social calculus. And I can attack these questions by using behavioral assays, measures of gene expression in the brain, and CRISPR-Cas9 gene editing. And this is what I've done ever since I became a part of the Arnold and Mabel Beckman Foundation, developing these tools more and more. And when I became a part of this foundation, I felt so inspired and at home, and to see how cherished he is at this foundation, and learn about these tips for success that when I looked at first, I said, "Oh, it's I've never really seen a scientist do this." It was sort of unexpected, um, but it's something that I really do stick to as I run my lab. And so, I find myself telling my lab, we strive for precision and clarity. A lot of that has to do with tip one. A lot of that has to do with the integrity and everything. Um, we as a lab, we balance our happiness. We need to do things in moderation. That means sometimes you may, including moderation itself, sometimes you may be in lab from 8 a.m. to 10 p.m. But once you're done with that subset of your work, you need to reward yourself. I love this one, hiring the best people and just letting them do their work and giving them what they need when they need it. Develop their independence, turn them into the best science they could be, and not being afraid to make mistakes. When people are learning, I tell them when I started out and when anyone started out doing this, there was a moment where they didn't know how to do this. It takes time, it takes focus, and it takes curiosity. And so these tips are really key for me. I use these as a mentor, as a scientist, and as a person trying to maintain balance in this, this pursuit of knowledge and happiness in life. So in my own lab as a Beckman Young investigator, I focus on wanting to develop these cichlids as a model for social behavior. And I did this using, in my proposal for the BYI, were three different sort of aims, and that was to use single cell and spatial transcriptomics, so the ability to label within cells hundreds or even thousands of transcripts to define what kind of cells they are. And then using the information about what is expressed in each cells to develop new models, new genetic models that lack function using CRISPR-Cas9 gene editing, sort of a genetic scissors. And finally, something that was really a focus of mine, to be able to eventually maybe call the NIH one day, is to develop a collaborative network of researchers to study social behavior in cichlids, to make it what mice are to scientists today, to make it a well-known model of social behavior. So what I want to do is just show you some progress of what we've done in the lab. We've used these single-cell transcriptomic approaches to understand the genetic markers in cell types in the brain of these cichlids. That is so important. In mice, what they've been able to do is use such precise information about the genes and different cell types to develop animals that lack the functions of genes in the brains throughout their body or specific cell types. And so just a brief overview is we've been able to parse out individual cell types within the social brain. The social brain includes the hypothalamus, which is a highly heterogeneous brain region. And here's some of the findings here that map back on to some important hormonal control of social behavior. We found that in glutamatergic neurons, a peptide called somatostatin that is normally involved in metabolism of growth is highly expressed in the dominant individuals. And here I'm showing you the expression of steroid hormone signaling genes. So these hormones that I showed you that were important, now we have an idea of where they're acting. They're acting within neurons involved in, or cells that are becoming new that are going to have to survive and incorporate to the brain, or astrocytes, the types of cells that support neurons and support the transmission of certain calcium influxes to regulate behavior. We're finding that these key genes involved in steroid hormone signaling are present there. And we're finding, really to our shock, very few of these genes, except for receptors for cortisol, which are stress hormones, very few of the neurons actually express these steroid hormone signaling genes. So we have surprising findings, findings that can guide our work to make new discoveries. And here's the spatial transcriptomics. This is something that I was the first to do, to look in the cichlid brain, take our data from the single cell RNA-seq and turn it onto the actual intact brain to see where these cells are. And so what you're seeing here is the ability of this program. It's called a Z-name analyzer. Once we have the tissue, we labeled 50 genes across the hypothalamus, using genetic markers to spatially validate what we found. And these are markers of astrocytes, and you could show here we are confirming the expression of these steroid hormone signaling genes, AR1, AR2, and this one's called CYP19A1B. These are the focus of this slide, that are expressed in these astrocytes. And so astrocytes being ones that are shown along sort of the midline of the brain. So we're confirming what we found. This is a huge, major step in understanding cichlid neuroscience and to develop these models further. We used the transcriptomics findings. Like I said, I mentioned somatostatin. Based on those findings, again, this has really transformed what we do. We developed a model based on those findings, and we found that in the early stages of developing them, the ones that actually lack the functional somatostatin gene, they're called a crispant, ones that were injected with the CRISPR construct. A mutated somatostatin means that they're interacting less during aggression and they're attacking less during aggression assays. So this is something that's very powerful for us to basically parse out. We found the genes different based on social rank. What does it do? So we engineered a mutant. And how are we doing on developing a collaborative network? Well, we think one step towards that is developing resources for the field. So this is showing an example here. Using our lab website, we are now creating what's called the Cichlid Brain and Behavior Hub, which has all of our projects available for other researchers to dissect. And so this is sort of mapping out. You can click through different steps here to gain access to all of our RNA-seq data of single-cell biology in collaboration with the cell browser at UCSC. And then you can look at different projects. What are the videos we use to analyze behavior? And people can go in here, validate what we did, replicate what we did, or use it for training in their own lab to just develop what are we doing, share it with the world to further develop what we're able to do and get what steps closer to developing this as a model of social behavior. So the conclusions here is that what we're doing, we think, is transforming how we understand social behavior mechanisms. We've published pieces of work on this, basically understanding whether genetics of social status, how are hormones playing a role differently based on whether the fish are males or females. So this is key information for us to know, and it's gaining attention, right? So we published one paper on these GnRH neurons showing that they're sensitive to androgen signaling, and this got the attention of the journal to a degree that our beautiful images were actually picked as the cover of the issue. the cover of the issue our paper was published in. So what we think we're doing here is we're showing the benefits of using cyclists to understanding brain and behavior. And next, what we're trying to do, we're going to use what we have from the single cell transcriptomics and spatial data to really try and gain more precise access to cellular control of behavior using techniques that allow you to manipulate genes in particular cell types. And then, of course, we need to plan that regional conference. So I am just so thankful to the Arnold and Mabel Beckman Foundation. They have been supporting me since 2017, and it's been huge for my career. It's been huge for me forming new networks, and it really has been inspiring. And so I want to thank the Arnold and Mabel Beckman Foundation for supporting me for all these years. And this is a bittersweet moment to be able to summarize my work thus far, but it's carrying me forward. It's keeping me inspired to inspire my team and perform inspiring work that I hope will really transform how we understand neuroscience. Thank you.